Good morning or good afternoon, and welcome to the Anemocyte talk. First of all, I want to thank the organizer to give us the opportunity to present our company. It's a pity that we can only meet virtual, and I really hope soon we can all meet in person. But in the meantime, I hope you will enjoy the presentation of today. The goal here is really to introduce the company, who we are and what we do, and to give you an example of what is our approach to the business in which we are in, which is the manufacturing of advanced therapies. First of all, it's important to stress that we are not a new company. We are actually in the business for long, not only as Anemocyte itself, but also as a member of a large families of companies, all focused in manufacturing products for the pharmaceutical industry. As you can see here, we are all under an umbrella name, the 9 Trees Group, and among other companies, we are trying to really deliver the best to the industry we serve. In this slide, we try to present our presence on a global scale. So as you can see here, most of the companies that are part of the 9 Trees Group are based in Italy or around Italy. However, we have also presence in North America and in Asia and Japan. Actually, the largest market in which the group is active is North America. Anemocyte decided to present itself to the business as a biotech manufacturing organization, which is a little bit an unusual way to define a company that is active in the manufacturing space. And so it might be interesting to explain why we decide to use this acronym rather than a CMO or CDMO that is more common in the space. So contract manufacturing means that there is a contract in place, and therefore that is the starting point of doing the business with our customer or collaborators. On the other end, we decide to define the biotech as really a mindset. So we are proactively looking into the challenges and bottleneck of the cell and gene therapy industry in order to be ready and to start with experience and capacity to guide a little bit the decision that our clients and the industry will require. So being a BMO implies really work proactively in addressing issue. And we do that in two main areas. One set, we are working on cell therapy, so working on autologous and allogenic product, non-modified, like mesenchymal stem cells, for example. We are also doing a lot in the non-viral space, and we are also working in plasmid manufacturing. So those are the two areas in which we are mainly active, and we have two manufacturing sites based outside Milan, where we manufacture the two products I just described. And the experience, well, that's also an important point. As a company, we have more than 15 years of experience, but we also leverage the experience of the group that, as I mentioned before, has been in the business for more than 60 years. Now, let me give you some example of what it means to be a BMO in action. And I think the best way is really to start from two keywords, that is manufacturability and sustainability. We did publish some poster and some position paper stressing that those two concepts, because for us are key in the cell and gene therapy space. And to better explain the concept of manufacturability and sustainability, that means to be able on one end to really develop a process that goes needle to needle. So from the starting material that sometimes is really a sample that comes from the patient to the delivery of the product back to the patient. So that's one side, the manufacturability, but you cannot just stop with the concept of, okay, I can do a drug. You have also to make sure it's sustainable. So it's important to look at the cost of goods, the regulatory requirements, and also the reimbursement. And that is really the surrounding of what are the four pillars then, that is the identity, safety, purity, and potency. And with this concept in mind, I will give you here a couple of examples of how we did approach that concept. So the picture here, I'm sure most of you are familiar with that, is just a cartoon summarizing how CAR-T are produced. This is the example in an autologous setting. So the leukapheresis is collected, and then there are a few steps to get to a final product. The first step is to modify the cells that come out from it. And the most common strategy is to rely on viral vector. I would say lenti is the way to go historically in the industry. But to produce lentiviral vector is not that easy. You don't find that off the shelf necessarily. You really have to think of what are the building blocks of it, and those are the plasmid. So there was an interesting article a few years ago presenting the fact that plasmid are a potential bottleneck of the industry. So we decided as a company to invest there. So in 2019, we did expand an existing manufacturing area for plasmid. Now is a full control and classify a series of rooms to guarantee the manufacturing of the product. And I don't want to enter too much into the details for the sake of time, but we have a state-of-the-art facility and manufacturing strategy in order to guarantee the right quality and at the right time for plasmid up to commercial application. The other aspect is, the production of the viral vectors. That's another area that was highlighted a few years ago to be foreseen as an area where there could be some potential shortage of manufacturing slot. Therefore, the viral vector was highlighted as an area of concern in the industry. I did try to summarize in this slide what are the main let's say red flag of viral vector. Most of them have been addressed in the year, so they're not really of a real concern. However, there is the manufacturing aspect and the cargo sides are still areas where the industry still see that as a potential challenge. So as an Anemocyte, we internalize this program. We look into possible solution, and we decide to invest into the electroporation based strategy. We did that starting a collaboration with Anemocyte, for which we are a center of excellence, and we did apply that technology together with some alternative strategies such as transposon, for example. Of course, the Anemocyte is a very flexible tool, so it does allow us to play a little bit around some of the different way to edit cells using electroporation rather than viral vector. Working as a BMO, it's as I mentioned in this slide, is not just playing alone. It means also to collaborate also with other company. And I want to share here one example is a collaboration that we had with Fujifilm last year, where we did use one of their media in comparison with an existing xeno-free and serum-free media. And the goal of the collaboration was really to test the capacity of this new media and see how that could impact our manufacturing strategy. And as you can see here, I mean, I'm also cutting a little bit some of the information, but what is key, you see on the left side, there is the cell number at different time points. So after 7 days, 10 days, and 14 days. And as you can see here, the red bar is the media that comes from the Fujifilm. And the numbers and the results were quite impressive. We've seen an higher expansion rate, 2 to 5 fold, while the viability was comparable between the two populations. We also have some interesting results in terms of analysis of the cells here. For the sake of time, I don't have the opportunity now to enter into much details, but what I want to stress is that by doing this test, we were able to see potential area of revisit the manufacturing approach to cell therapy up to a point where we could better either obtain more cells or results in saving in terms of reagent that are used, such as cytokine. And we think this is the right example to explain really how our approach as a BMO will result in potential knowledge that can result as an advantage to the company that works with us. So in conclusion, we are a company that is very much active in the manufacturing side, but we also have a team of people that is really looking into possible solution and option so that we can really working in a working as a consultant somehow in driving some of the decision, and make sure even early in your the manufacturing stage, you can take the right educated decision. Of course, every manufacturing story have its own nuances, so it's very important to be able to identify the right sweet spot for both manufacturability and sustainability, and several forces enter into play. So every new product have its own story. Again, I want to stress the fact that our focus for development manufacturing is the cell therapy and plasmid. We do that as of today mainly in the facility you see here in the picture. Again, we have the 2 manufacturing sites in the V-shaped building, and we are growing as a company. So we hope in the coming years to be able to share more news in terms of increasing of space and also new activities that are coming up in our site. So this is my last slide. I want to thank you for the attention. And of course, if there will be any question, you can easily reach out to me through the partnering system. Thanks a lot.